In this lecture, we will look at regulation of the citric acid cycle and also a little bit later into citric acid cycle as source of biosynthetic intermediates and finally into one related cycle, glyoxalate cycle. First point of the regulation of the citric acid cycle is control of the entry into the citric acid cycle. And that means regulation of the pyruvate dehydrogenase complex. It's accomplished both allosterically and by reversible phosphorylation or covalent modification. In animals, conversion of pyruvate into acetyl coenzyme A is an irreversible process as they are not able to convert acetyl coenzyme A into glucose, which means that once acetyl coenzyme A is formed, that commits carbon atoms of glucose either to oxidation to carbon dioxide by the citric acid cycle, and that means generation of energy, or incorporation into a lipid. Lipid is long-term energy storage. So this is critical branching point of metabolism, and for that reason, activity of pyruvate dehydrogenase complex is strictly controlled. High concentration of the reaction products inhibits the reaction. So acetyl coenzyme A inhibits trans-acetylase component E2 by direct binding, and NADH inhibits dihydrolipoyl dehydrogenase E3. The key means of regulation is covalent modification of the pyruvate dehydrogenase component E1 by reversible phosphorylation. Phosphorylation is accomplished by specific kinase enzyme that switches off the activity of the complex, and the activation is reversed by the action of the specific phosphatase enzyme. These two enzymes, kinase and phosphatase, are regulated by the ratios of NADH to NAD, acetyl coenzyme A to coenzyme A, and ATP to ADP. High ratios indicate that there are no significant energy demands, so they promote phosphorylation and deactivation of the pyruvate dehydrogenase complex. As ADP and pyruvate accumulate, they activate the pyruvate dehydrogenase complex by inhibiting the kinase enzyme. And so then phosphatase becomes active, dephosphorylates inactive PDH, and PDH becomes active. PDH is pyruvate dehydrogenase complex. Another way to control the citric acid cycle is to control its rate. Primary control points are the first two enzymes that generate high energy electrons, and those are isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. So when it comes to regulation of isocitrate dehydrogenase, the mechanism of regulation is the reverse of regulation of pyruvate dehydrogenase. It is allosterically activated by ADP and NAD+, and inhibited by ATP and NADH, which means that high NAD+ and NADH and ADP-ATP ratios stimulate isocitrate dehydrogenase and citric acid cycle activity. Binding of isocitrate, NAD, magnesium ions, and ADP is mutually cooperative. Inhibition of isocitrate dehydrogenase leads to buildup of citrate, which is then transported into cytoplasm, where it signals phosphofructokinase to stop glycolysis, and where it can serve as a source of acetyl coenzyme A for fatty acid synthesis. The other enzyme that is regulated is alpha-ketoglutarate dehydrogenase. It is homologous with pyruvate dehydrogenase, and some aspects of control are similar. Alpha-ketoglutarate dehydrogenase is inhibited by the reaction products and a high energy charge. So succinyl coenzyme A, reaction product, NADH, and ATP, indicators of high energy charge, are all inhibitors. Alpha-ketoglutarate that accumulates when alpha-ketoglutarate dehydrogenase is inhibited is actually a rather useful compound and can be used as precursor for several amino acids and purine bases. So it could be drawn out of the citric acid cycle and used as precursor for synthesis of biomolecules. Besides synthesis of ATP, or energy, and electrons for the oxidative phosphorylation, citric acid cycle also provides intermediates for biosynthesis. For example, succinyl coenzyme A is used in synthesis of porphyrins, and alpha-ketoglutarate and oxaloacetate are used in synthesis of amino acids. If used in biosynthesis, those intermediates are drawn off the citric acid cycle and must be replenished. Oxaloacetate is generated in carboxylation of pyruvate. We have actually seen that when we studied gluconeogenesis. Pyruvate carboxylase is active only in the presence of acetyl coenzyme A, which indicates a need for oxaloacetate. Because citric acid cycle is a cycle, so if intermediates are drawn off for biosynthesis, cycle can be replenished by generation of any other intermediate. Anaplerotic reactions are reactions that generate intermediates in metabolic pathway. If intermediates are drawn off for the citric acid cycle, from the citric acid cycle for biosynthesis, the cycle can be replenished through a process called anaplerosis. That means generation of citric acid cycle intermediates. The most important citric acid cycle anaplerotic reaction is generation of oxaloacetate from pyruvate. And finally, we are going to look at example of another metabolic cycle other than the citric acid cycle, and that's the glyoxalate cycle. Glyoxalate cycle is a truncated version of the citric acid cycle. It converts acetyl coenzyme A to biosynthetic intermediates without loss of carbon dioxide. So, this metabolic pathway is essentially a modified version of the citric acid cycle that bypasses steps that lead to loss of carbon dioxide. That's shown on the diagram below. So in dashed ellipse, those steps that are bypassed are indicated. And in bold arrows, glyoxalate cycle is shown. This pathway does not occur in vertebrates, but it is found in plants and certain bacteria, fungi, and invertebrates. The glyoxalate cycle is a sequence of anaplerotic reactions that enables an organism to use acetyl coenzyme A as the sole carbon source. The glyoxalate cycle utilizes a two-step bypass. One key enzyme, isocitrate lyase, converts isocitrate to succinate and glyoxalate. So at that point, cycle bifurcates to 2-carbon unit glyoxalate and 4-carbon unit succinate. Second key enzyme, malate synthase, condenses glyoxalate, 2-carbon unit, with molecule of acetyl coenzyme A to form malate. And of course, in ordinary citric acid cycle reactions, succinate is also converted to malate, and then cycle continues from malate to oxaloacetate, citrate, and so on. So the succinate that was formed by isocitrate lyase can be withdrawn from the cycle and used for cell carbon biosynthesis. Some organisms also possess other alternative pathways that convert acetyl coenzyme A to 4-carbon biosynthetic intermediates, and those would be ethyl malonyl coenzyme pathway and methyl aspartate cycle, just to name a few.